right, I'm back. So let me talk about these four, and then we'll talk about the quiz. So let's talk about cells first. So as you can see, there's two types of cells. They're phagocytes, and phagocytes are going to they're going to surround the pathogen and then take it in and eat it. I'm going to post a cool video of like a neutrophil eating what virus was it? Strep. I'll show you a photo of a neutrophil chasing after a streptococcus bacteria and eating it. It's kind of cool. So some of them are just wandering around, like the one that I'll show you in the video, and then some of them are fixed. They're like underneath the epithelial tissue, and they're just waiting. So that's one way for a cell to engage a pathogen. But the other way is to attack it directly. And so this is there's natural killer cells in your body. They're actually very similar to T8 cells. It's just that they use the same weapons, right? And these are the two weapons that they have at their disposal. One's called perforin. And if you think about perforated paper, what is perforated paper? It's got a bunch of holes in it, and so you can tear the paper at the holes. That's what perforations are, like little holes. And that's what natural killer cells can do. They can release a protein called perforin, and it punches holes in the pathogen and makes everything like leak out. So we call that cytolysis or cytolysis. So you're breaking the cell open, and yeah, that's perforin. The other one is the use of granzymes. So I've got this word here, granzymes induce apoptosis. Apoptosis is cell suicide. So we have this thought that cells have like a pre-programmed time of death, where they're just going to kill themselves. So what granzymes do is it'll tell the cell, hey, that time that you got built in to kill yourself, that time's now. You got to kill yourself now. And so that's what I mean by inducing apoptosis. So that's the two weapons that natural killer cells use. They're going to directly confront the pathogen and like stab it with perforin, or they tell it to kill itself. Right? Whereas macrophages or phagocytes, they just surround the cell and eat it. So those are the two cells. But we also have proteins. And so something I want to tell you before I do that slide. So there's two types of pathogens. When we look at pathogens, we can look at them as exogenous. And that means that they attack the cell from the outside. And that's pretty standard, right? But viruses are a little bit different, and we call them endogenous. They attack from the inside. So endogenous are viruses. So here's like the cell. Here's the nucleus of the cell. And what viruses do is, all viruses is DNA or RNA, either one, DNA or RNA. So I'm just going to write this as I'll do DNA. I can't write that right. DNA surrounded by a protein. They're not even living. It's DNA surrounded by a protein. And what they'll do, this is like one thing that viruses can do. When they get up to the cell, they're going to inject the DNA into the cell. So here's my DNA strand. That one looks better. That DNA has instructions on how to make more of it. So that DNA is going to go into our nucleus and hijack our DNA, and they get our cells to make more viruses. So our cells make a bunch of new viruses, and then they're going to leave and go attack more cells. So that's how viruses work. They're like a Trojan horse. You don't know that they're there until they're inside. They're really good at camouflaging themselves. So they'll, like on their way out of our cell, some of them will surround themselves with our membrane. And so when you look at it, it looks like us. And then they go attack other cells. So the thing is, viruses, if we could somehow have advanced notice that they're around, then we could do something about it. But the problem is, we never know they're there until they dock on our cell and put their DNA into us, or RNA. So we don't know that they're there until it's too late. So interferons can be released, these proteins can be released to the cells around it, saying, hey, look, it's too late for me, but you have a chance, make your antiviral proteins. We could make antiviral proteins if we just knew that we were infected. That's kind of what interferons do. You remember transferrin when we were talking about blood. The transferrin was transporting iron. Some bacteria that are pathogenic to us, they use iron to grow. So if you could take that iron away from those bacteria, it makes it difficult for them to grow. And then complement is another protein that's going to enhance antibodies, like help antibodies do their job, and they help phagocytes do their job. So these are like 3 proteins, antimicrobial proteins. That's another type of second line of defense. The third and the fourth are right here. Inflammation and fever. I kind of did this wrong. It's inflammation's here, this bullet point. Fever is that bullet point. Fever, like I said, it increases your metabolism, makes things hotter. Microbes or pathogens don't like that temperature. But vasodilation is 2 things. One, you increase the permeability. You make it easier for things to get outside of the capillaries. Like you're making filtration a lot easier. That's what increasing permeability is. Right? And then you want dilation. You want, which I spelled it right, you want your blood vessels to dilate, and because you want the blood to move faster through it and get to where it has to go. And then you want like white blood cells to get out of the blood, out of the capillary, and into the interstitial fluid. And that's why when you have like allergies, you're feeling like runny nose and like, I don't know how to describe it, but you know what I'm saying. Right? Because that's what's happening. You're getting a bunch of inflammation in your face, sinuses and stuff. So there's some chemicals that are involved in this too, and you've probably heard of them. You've heard of histamines, right? So they're gonna induce vasodilation. So that's fine. And then kinins do the same thing. So antihistamines like also antikinins, that's kind of going against this. You don't want your blood vessels to be dilated like in your eyes and nose and stuff like that, because that's going to also cause like increased permeability. And then prostaglandins are going to help with that. So you've got kinins and histamines that are helping with vasodilation. You have prostaglandins that are also helping those two, the histamines and kinins. And then you have leukotrienes, which increase permeability of the blood vessels. So that's inflammation. You want to kind of trap the pathogen or whatever it is. You want to trap it there. I was using the bee sting as the example, right? You want to trap it in your hands. You want to get a bunch of fluid in there and just make it really crowded and trap it. But then you also want to deliver like white blood cells and some proteins to that area to kind of solve the problem. We're going to cover this in next class. What I'll spend the rest of this time doing is going over things that things that I might put on the quiz. So we just had an exam, right? What did we just have a quiz? It was just on the heart and blood. We just had an exam, right?
Yeah.
Huh?
Yeah, we just had an exam. This would be, I think, quiz 3.
So I'm thinking just on blood vessels. So let's talk about some of those questions. There's 3 types of capillaries that we talked about: continuous, fenestrated, sinusoidal. I'd like for you to just briefly say what those are. Like, what's the difference between like a fenestrated capillary and a sinusoidal capillary? So, it all had to do with the intercellular clefts, and then in the case of the fenestrated, they had these pores on them, fenestrations, and sinusoid just had even larger clefts and larger fenestrations. And I mean, that'd be good enough. So whatever you put down for fenestrated, you can even use the same words and just put a lot bigger fenestrations and intercellular clefts. Continuous, which is the first one, had just small intercellular clefts. That's it. That's the only way that things can leave that type of capillary. In no particular order, what are the pressures driving filtration and reabsorption? Reabsorption. So filtration is fluid leaving the capillary. Reabsorption is fluid coming back into the capillary. There were 2 pressures driving filtration. So you have to describe them. Blood hydrostatic pressure. What do you mean by that? And then interstitial fluid osmotic pressure. What do you mean by that? And then blood colloid osmotic pressure. What do you mean by that? It's the albumin. More albumin in the blood colloid in the blood than in the interstitial fluid. But anyway, we talked about that. So that's 2 questions so far. Difference between muscular and elastic arteries. So what's the difference between muscular and elastic arteries? And it all has to do with that tunica media, the middle coat of the artery, right? So with elastic arteries, which are known as, I forgot the name, conducting arteries, the elastic part of that middle layer is thick. And with distributing arteries, muscular, the muscle layers of the tunica media is thick. So, again, I'm in no order here. What are the 4 types of hyperperfusion? Hypoperfusion, aka shock. What are the 4 types of shock? We didn't talk about every type, but we talked about 4 types. So, like, obstructive and vascular, and what's happening. With obstructive, it's kind of easy, right? There's an obstruction there. With cardiogenic, the heart's not functioning properly. Vascular is inappropriate vasodilation. And then whatever the other one is that, hypovolemic, not enough blood volume. Like you should know what it is. Like you should be able to say a couple of words about what it is. Don't put PEMI. I know you know how to remember letters. Just, briefly say what it is. And then how can we control for it? How do we compensate? So you can talk about like epinephrine, antidiuretic hormone, aldosterone. Our own nervous system can cause our blood vessels to, vasoconstriction from the sympathetic nervous system. Thinking control of blood pressure. So they're just like heart rate. There is a neurogenic control, like a nervous system control. The receptors, proprioceptors and baroreceptors. Well, not so much proprio, but baroreceptors, chemoreceptors. And then that your cardiovascular center can stimulate your blood vessels and cause them to constrict or dilate, depending on the situation. Right, and then there's hormones. Again, these hormones here are going to be the same ones for here. The only exception is atrial natriuretic peptide, ANP, is going to decrease blood pressure. So whatever you know about vasopressin, about antidiuretic hormone, it's the opposite. Whatever antidiuretic hormone does, this does the opposite. So that's going to make blood pressure go down. And then I had one more that I can't think of. Oh, yeah. What are the factors contributing to resistance? There were 3. The length of the blood vessel, the longer the vessels are, or the more vessels you have in your body, really, the more resistance you have. 2, the viscosity of the blood. The thicker that the blood is, the more resistance. So we call that viscosity. 3rd, the diameter of the lumen. So the smaller you make the diameter, the more resistance you have. And that's just like, what happens when you take a 4-lane interstate and you cut off just one lane, right? Everything backs up behind it, especially in Louisiana. Maybe it was here and then she got out. I'll pick 5 of those questions. I can't think of anything else. Do you want to put the functions of the lymphatic system on there, or do you care? I'll leave it. These are your 6 questions. I'll take 5. And that's it. If you don't have any questions, I'll see you here on Wednesday and we'll finish up the immune system. I didn't want to get too into it since we weren't doing the quiz on it, but I had to make some progress today as well. So there we go. I'll post this in about an hour or so. Bye. Bye-bye.